So, welcome everyone to this first seminar of the Kalim Redtalks sessions. I am Bertrand Nedel, director of the Kalim Institute. So, you're all here to hear our speakers. So I'll be brief, just a word to bring this first Red Talk in context. First, for our external guests, because I know that some of you are connected and do not really know and are probably wondering who we are. So what is Kalim? Kalim is a French consortium dedicated to basic and clinical research on lymphoma. It brings together French experts on lymphoma, research labs, clinical centers, cooperative groups, and professional structures of clinical research. And together we provide an integrated expertise allowing to connect the segment of the research and development chain, discovery, innovation, transfer to rationalize biology-based trial design, and to interface the know-how and best practice of the academic and industrial world. So Kalim is now credited since 2011 with the Carnot Institute Label of Excellence for professionalized partnership with industry, delivered by the French Ministry of Research and Technology. If you want to know more, please visit our wonderful website. So now, what the heck is a Red Talk? With the COVID, we all learned to adapt. And some of us, I was one of them, experienced our first webinars. And I was myself very surprised actually to see how much I enjoy talks in my living room with a drink. Actually, I have one, it's tea. I guess this was part of the reason why the webinar started to gain some large audience. So we thought, well, it might be really nice to have a regular scientific event focused on lymphoma hot topics, which should bring together experts from the Kalim Consortium and beyond. We see it together today to foster discussions, brainstorming, networking, collaborations, and so on. So we decided to create the Kalim Red Talks, this webinar series focused on lymphoma. So the Red Talks will be monthly, every first Thursday of each month at 6 p.m. Central European time, so that we can accommodate a maximum of other time zones. We have today some people from Europe, from North America, from Africa, and many thanks to you to join us in this webinar. This is an open webinar, it's absolutely free, you just need to register as you did today, and that's it. And if you glance at the upcoming program, you will see that we have international stars on lymphoma, starting today with Jean-Ehrland. Many thanks in advance to Jean-Ehrland, RICCI today, and all program speakers for accepting our invitations. And last but not least, a bit of standard operating procedure on the LiveStorm platform. It's a webinar, so you won't be able to directly ask questions to the speaker at the end of the talk. But you have a wonderful moderator today, François Lemonnier, who will do it for you. Just formulate your question through the question tab on your right, and you will have the possibility to vote even for a question you believe should absolutely be asked to the speaker. And finally, you will also have the possibility to chat with the audience. And for this, please use the chat tab. So that's it. I'm done. I don't want to take more time. I want to have the room for Jean-Ehrland as fast as possible. I hope you will enjoy the first Red Talk, and François Lemonnier will share the session. François, the floor is yours.
Thank you very much, Bertrand. So this is my great pleasure to introduce for this first Kalim Red Talk Jean-Ehrland RICCI, who will talk about sugar and lymphoma. So Jean-Ehrland received his master's degree and his PhD in molecular and cellular biology at the University of Nice, Sophia Antipolis, where he studied fast and T-cell receptor-mediated apoptosis in human T cells. Then, from 2000 to 2004, he joined Dr. Douglas Bruin Laboratory in San Diego for his postdoctoral training, where he studied the metabolic regulation of caspase-dependent and independent cell death. Since 2006, he leads the Cancer Metabolism and Immune Response team at Inserm U1065 in Nice, France, and his research focuses on how metabolism and metabolic enzymes control signaling pathways, cell death, and immune response in lymphoma, going from preclinical models to clinical studies. So recently, his team published two important papers in cell metabolism and cancer cells exploring the role of GAPDH in DLBCL and in T-cell context. So I will not scoop this unexpected effect of GAPDH and let Jean-Ehrland RICCI present his very interesting results. So Jean-Ehrland, please.
Thank you very much, François, and thank you, Bertrand, for this invitation. It's really a great pleasure to be the first one to initiate this Redtalks from the Kalim Institute. And today we're going to talk about the link between metabolism and lymphoma. And I would like to mention that my team is part of Inserm, University Côte d'Azur, and I'm also part of the Scientific Council of Canceropol PACA that help us for our projects. So why should we care about metabolism? It has been shown about a century ago by Otto Warburg that the metabolism of tumor cell is different than the metabolism of most normal cells. And what Otto Warburg described is that tumor cells have enormous avidity for glucose. And this glucose is then converted to lactate, and this regardless of the oxygen availability. So the dependence of tumor cell on glucose led the field to initiate the notion of the TEP scan technology, and we know how important it is for the treatment and the diagnosis. But since the discovery of Otto Warburg, of course, we now understand better in a more global picture how metabolism may be impacting on tumor cell physiology. And as opposed to what Warburg thought, most of the glucose is still converted to pyruvate, but pyruvate is used by the mitochondrial to produce energy through the TCA cycle mainly. And actually, it has been shown that mitochondria are absolutely necessary for tumor growth in most cases. So not only glucose and mitochondria can produce energy, but also lipids is a very important source of energy in the cell. And through its import or lipogenesis, that will feed, for example, the TCA cycle. So when we look into more details, don't get afraid. I won't guide you through all of those metabolites yet. But the idea of this slide is to show the connection between the different metabolic pathways. So between glycolysis, you may have connections to lipid biosynthesis, nucleotide biosynthesis, amino acid synthesis, that will all of them somehow directly or more indirectly feed back to mitochondria. So it's extremely connected, and the intermediate of the glycolysis are probably as important as the energy produced by glycolysis itself. So glycolysis should not be seen as a tunnel where glucose gets on one side and pyruvate on the other one. Of course, energy is part of it, but we just saw that the building block arising from glycolysis are very important for cell proliferation, and not mainly in the context of cancers. But also different metabolites or intermediates or enzymes of this pathway can control immune evasion, signalings, and transcriptional and translational regulations. So with all that in mind, and the notion that this metabolism of tumor cell is different from a normal cell, this led the field to develop metabolic inhibitors with the idea to target the specificity of the tumor cells. And as we speak, there is about 100 clinical trials that are ongoing, combining different kinds of metabolic inhibitors with standard of care in a wide variety of cancer treatment. But unfortunately, as far as we know, only a handful of clinical trials gave a benefit to the patients. And this is extremely disappointing. And there is a few reasons that could explain that. One of the main reasons that is raised is, of course, inhibiting metabolism. And actually, for example, inhibiting a limiting enzyme of a metabolic pathway is extremely toxic because, of course, a lot of cells, like the brain, will eventually use glycolysis, for example, to proliferate in normal conditions. So toxicity can be a main drawback, but of course it can be managed according to different molecules or some therapeutic windows. Another concern that is often brought in the field is the bioavailability of the compound within the tumor. That is often said for metformin or derivative of metformin that are somehow inhibiting complex 1 of the electron transport chain. And people are arguing that you may not be able to have enough amount of it within the tumor in the proper environment. And of course, it's a huge debate in the field. But there is two notions that are often underappreciated and that also participate in this disappointing benefit effect of the use of metabolic inhibitor. And the first one is the enormous dependency on those study of the use of cancer cell lines. So you have to realize that the cell lines were isolated decades ago. The first one was the HeLa cells in 1951. And since those cells are grown in high glucose, high amino acid, high oxygen contents, medias, and this is very likely to completely modify their metabolism. So we have an enormous dependency of cell lines on glucose and glutamine, but this is not necessarily happening in real life settings. Another thing that is also a drawback in this field is the injection of cell lines in subcutaneously in immunodeficient mice. It's a drawback for two reasons. First, because those cells will be put in a non-physiological environment, meaning they will go through enormous restriction of oxygen and nutrients in the first step of the growth of the tumor. And the other things is you cannot appreciate the role of those metabolic inhibitors on the immune cells, and we know how important it is, and we'll discuss this point in the second part of this presentation. Another concern that is often underappreciated, and the notion is quite novel, it's like 15 years ago, that has been described that within the same tumor entity for a given patient, and sometimes with the same genetic alterations, some patients will develop a so-called OXPHOS tumor, and other patients will mainly develop a tumor using their mitochondria, so-called oxidative phosphorylation or OXPHOS tumor. So of course, if you take these kind of patients and you target, for example, glycolysis in this context, it's very unlikely to give any benefit to the patient. And so far, we have no way to discriminate within the tumor entity which type of tumor a patient is developing. So with that in mind, we have, I'm going to tell you two of the recent stories we have made in my lab, and one is how can we optimize the use of those metabolic inhibitors, mainly in B-cell lymphomas. And in the second part, we'll discuss the importance of glycolytic enzyme in T cells. So we are mainly studying diffuse large B-cell lymphomas, and I will refer to it as DLBCL. That is the most frequent and account for most of the aggressive form of non-Hodgkin lymphomas. It has a very profound dysregulated metabolism, and the incidence of this lymphoma increases a lot with aging, upon aging. So as we speak, the standard of care is still a combination of immunochemotherapy combining anti-CD20 antibody, rituximab, and CD20 is expressed during the differentiation of B cells with a cocktail of 4 chemotherapy, and is referred as R-CHOP. So when a patient is diagnosed, he received R-CHOP, and about 40% of those patients will somehow relapse or be refractory to the treatment. Then patients will be switched to another cocktail of chemotherapy combined to anti-CD20, and here again, about another 40% of patients will fail, will finally not respond to the treatment. And if they are eligible, and for some of them, they will benefit from the CAR T cell therapy, but we know that probably 50% of those patients will relapse from the CAR T cell therapy, and the other patients will go to clinical phase or eventually to supportive care. So a few years ago, a talented researcher in my lab, Jean-Ehrland, asked the question, what could be different at the genetic level between patients having a good response and patients having a poor response to R-CHOP treatment? And I would like to mention the different foundations and associations that supported our work about over the last years, and mainly the Fondation ARC pour la Recherche Médicale. So by doing so, and by analyzing about 300 patients, patient samples, Joanna could identify more than 500 genes that were differentially expressed between high and low responder to R-CHOP. But among those genes, we get interested in one of them called GAPDH, And one of the reasons why we got interested in GABDH, because it was the only enzyme of the glycolytic pathway represented here by the 10 different enzymes, that was the only enzyme of this pathway that was predicting the response to R-CHOP. So it looked like this. So as you can see in black, patients expressing low level of GABDH at diagnosis have a poor prognosis compared to patients expressing high level of GABDH. So what is GABDH? So it encodes for glyceraldehyde 3-phosphate dehydrogenase that converts glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate, leading to the regeneration of NADH. But on top of his day job, GABDH has numerous night jobs, so-called non-metabolic functions. It could, for example, participate in cell proliferation, evasion from cell death. It can be controlled by the redox status of the cells. And we have shown over the years that it can control key signaling pathways, including NF-kappa B or mTOR. So no, GABDH is not a nice housekeeping genes. And to further emphasize this, we set up in collaboration with Professor Nikiels and Julie Reverso an automated EHC immunostaining for GABDH. And as you can appreciate, those are samples of patients at diagnosis, and some patients will have low expression of GABDH, and some patients will have high expression of GABDH. And we validated that in about 350 patient samples in collaboration with Professor Thierry Molina at the LISARC. And we found that indeed about 40% to the patient will be GABDH low, while the remaining 50, 60% of the patient will be GABDH high. And this correlates with the prediction that GABDH low patients, as we saw at the genetic level, but GABDH patients expressing low level of protein of GABDH respond poorly to R-CHOP treatment. So we did an univariate and multivariate analysis in both cohorts to show that GABDH is indeed an independent biomarker of the response to R-CHOP, and those are the clinical marker used as we speak. So we end up in a funny situation. We have a glycolytic enzyme supposed to be ubiquitous, but actually it's not. Some patients at diagnosis express low levels, others express high levels, and that predict very nicely the response to the standard of care. So as you may probably know, DLBCL is extremely heterogeneous, and it has been shown first about 20 years ago at the molecular level, and now we know more precisely by very tremendous work of different teams that there is a different wide range of pathologies of molecular heterogeneity within this DLBCL entity. But in parallel of this molecular heterogeneity, Margaret Chip and collaborators showed that there is indeed this metabolic heterogeneity I was referring to in my introduction. So some patients will develop glycolysis type of tumor, and other patients will develop oxidative type of tumor. But it has been shown also that there is no correlation between the metabolic and the molecular heterogeneity of those cells. So we then made the following hypothesis. As we have patients with high or low amount of GABDH, could it be a predictive marker of the metabolic status of the patient? And of course, the cohort of interest, the patients of interest, are the one poorly responding to R-CHOP, which are GABDH low. And if GABDH low are OXPHOS tumors, then we could envision to target mitochondria specifically using some of the inhibitors that has been developed. And some of them are readily available, they are on the market and used in clinic. And we could then target specifically this type of patients. So this is what we tested. To do so, we first used a primary model of non-Hodgkin B lymphoma, the immunic mice generated by Jerry Adams. And as you can appreciate, so those are primary sample of the mice. GABDH low lymphomas will produce less lactate, will consume more glutamine, and will respire more than the glycolytic tumors. So we did a more thorough analysis of the metabolism of those cells, and we observed that the prediction was true, meaning the GAPDH high have an accumulation of glycolytic intermediates, while GAPDH low tumors, and those are primary tumors isolated from the mice and directly snap freezed and analyzed by LCMS, GAPDH low cells have an OXPHOS accumulation of OXPHOS intermediates. And finally, it's not only true in mice, it's also true in humans. When we analyzed about 150 patients, we found that OXPHOS patients have significantly less GAPDH compared to glycolytic patients. So can we target then this metabolism, and more specifically the metabolism of the mitochondria? So we used a variety of inhibitor, and the first one I'm going to present to you is the fenformin. So fenformin is an analog of metformin that is a mitochondrial complex 1 inhibitor. And we could show ex vivo on primary lymphoma cells first that the glycolytic tumors do not respond to fenformin as expected because they are not using their mitochondria to produce their energy, while the GAPDH low cells are very sensitive to the cytotoxicity induced by this compound. So we then, of course, validated this result in vivo. And when mice bearing OXPHOS lymphomas are treated with fenformin, they do respond to it. On the opposite, when the mice are developing glycolytic tumor, even though they do share the same genetic alteration, glycolytic tumor do not respond to fenformin. So another inhibitor we used was the L-asparaginase. That is an enzyme very efficient that efficiently removes asparagine but also glutamine from the sera and then in the tumor. And as we saw before, OXPHOS tumor are poorly sensitive to L-asparaginase, while glycolytic tumor are poorly responding, while OXPHOS tumor are very sensitive to it. And it's even more striking in vivo when when mice bearing OXPHOS lymphoma are really nicely protected with the use of L-asparaginase, and it is not the case when mice are developing glycolytic tumors. So of course, if you make a clinical trial mixing those two populations, because you have no way to discriminate it, it's very likely part of the reason why you cannot find beneficial effects in trials. So the take-home message of this first part is this glycolytic enzyme GAPDH turned out to be a therapeutic factor for DLBCL, as it can predict the response to standard of care, but also allow for the first time to know which is the metabolic status of a given patient, and then eventually offer therapeutic option to those patients. So in my lab, as you figure it out, we are doing fundamental research, and in the field, it's extremely complicated to go from fundamental research to clinic, and this is often referred as the valley of death, where it has been shown already about 10 years ago that more than 150,000 biomarkers are claimed to be present in PubMed, but less than 100 are really used in clinic. So we wanted to know how robust GAPDH was as a predictive marker, and we were extremely lucky and happy to have a nice and a wonderful collaboration with Professor Catherine Thieblemont, who is the head of the oncohematology department at Saint-Louis Hospital in Paris. And Catherine was facing sometimes those patients that do not respond to any of the treatment that they could offer. And this is an example of it. The first treatment, as you can appreciate, a very aggressive form of lymphoma and very visual on his face. So retrospectively, we could analyze the sample, the initial at the diagnosis, the initial sample of this patient, and it turned out that this patient had a GAPDH score of 35. Just a reminder, the cutoff between high and low levels of GAPDH is 150. So this patient at diagnosis had very, very low level of GAPDH. That is in line with the fact that this patient was not responding to standard of care. So what Catherine and the different clinicians in association with the LISA Association decided to do, it was to combine a set of metabolic inhibitor, meaning hydrolase, every 2 days for the first 2 weeks, and then they had to stop hydrolase, and they decided to put metformin to continue to inhibit mitochondrial functions for a while. And hydrolase was put in association with the rapalog tensirolimus. So the first patient we saw before was treated with this treatment for a few cycles. And this is the same patient after 7 days and the same patients after the end of the first cycle. He had a complete response, a decrease of the tumor mice by more than 80%, and actually was negative at TEP scan and had a normal life for several months. So this is not only a case report. Catherine and the clinicians could offer this compassionate protocol to 9 patients. And actually it turned out that 7 out of those 9 patients were indeed GABDH low from diagnosis. So confirming that we could predict that those patients would be poorly responding to the treatments. And only 4 of those patients could benefit from 1 cycle of a KTM protocol, and 3 out of those 4 patients were in complete responses and could extend their life for a few months. So obviously, as you saw, this is a teamwork between fundamental researchers in my lab, but also in different places in France, but obviously also with clinicians and bioinformaticians that help us to combine and to identify a new marker, not only to predict the response to R-CHOP, but for the first time to be able to estimate the metabolic status of a tumor that we think is absolutely required before using any metabolic inhibitor in patients. So the second part of my talk is dealing with the role of glycolytic enzyme in T cells. And this work was made by Laura Mondragon, Rana Emendli, under the supervision of Els Verhooyen in my lab, and supported by the different foundations and associations presented below. So as you may know, during T cell activation, they do go through this so-called Warburg effect. Naive T cells are mainly using OXPHOS, while activated T cells are mainly using glycolysis. And it has been shown that this switch in metabolism is absolutely crucial for the proliferation, but also for the functions of those T cells. So for a lot of reasons I don't have time to explain to you right now, we thought that GABDH, having more GABDH will make a difference on the proliferation or the functions of those T cells. And actually, GABDH is mainly overexpressed, for example, when you are in hypoxic conditions, and the thymus, where thymocytes are developing, is one of the most hypoxic area of the body, most hypoxic organs, where you have an enormous increase in GABDH. So to know specifically what an increase of GABDH in T cells could do, we generated the mice, overexpressing it under the LCK promoter, and we have 3 different founders. And it turned out that upon aging, when they turn about 18 months of age, all the mice will at some point develop a lymphoproliferative disorder associated with a splenomegaly and an enlarged lymph nodes. So when we characterize the T cells in those mice, it appears that we have an enormous proportion of CD4 over CD8, and a further characterization could tell us that we have, with the characteristic described here, an increase in T follicular helper in those mice. So as you may know, those particular type of T cells reside within the germinal center and interact with the B cells to allow their maturation and functions. So we didn't find an increase in the number of B cells in those mice, but we do find an increase in the proportion of JCBs in those mice. So the summary of the phenotype of those mice is an increase in T follicular helper, a strong inflammatory phenotype, an enormous T cell proliferation, a gamma and beta clonality of the T cells, an increase in JCBs, as we saw, germinal center B cells. And of course, we could transfer those tumor cells to a recipient mice and transfer the lymphoma in another mice. So with all those clinical or characteristic of those mice, it turned out to be very closely related to a rare human disease called Angioimmunoblastic T-cell lymphoma, or AITL. So AITL is a type of non-Hodgkin T-cell lymphoma that accounts for 20 to 15% of all T-cell lymphomas, and it is characterized by a dominance of T follicular helper with a strong B-cell component. And as we speak, there is no specific therapies for this rare disease. So when we made a step-by-step comparison of the clinical feature observed in human AITL, we observed a very, very nice correlation with the characteristic in mice, with skin rashes, ascites, splenomegaly, proliferation and clonal TFH, interaction of TFH and GCBs, bone marrow infiltration, and the ability to transfer those tumor cells into a recipient mice. But AITL is characterized also by genetic alteration. So there is a variety of them, but one of them is very predominant, as from 50 to 70% of the patients will bear a mutation, a G17V ROA mutation, that is a loss of function mutations. So we did a Sanger sequences over mice, and actually it turned out very surprisingly that all the mice we analyzed, we sequenced, presented another type of the ROA mutations, and this mutation is the T37M that is within the effector domain, and we could characterize that this mutation is indeed the loss of function, as this point mutation prevent the ability of the protein to interact with Rho-GTP. So indeed, to our big surprise, this PLCK-GAPDH mice to be a very nice and closely related model of AITL. So of course, a question that keeps us busy for quite a long time, to be honest, is how a glycolytic enzyme can do that. How is that possible? Well, we looked a lot for the answer, but finally the solutions came from a work that Joanna did actually a few years ago in B cells. And what Joanna could show in this in those cells is that GAPDH has the ability, when you overexpress it, when you have more GAPDH in a cell, it has the ability to bind STRAF2, and that will lead to an increase of the NF-kappa B pathway. So that's indeed what we observed in the thymocytes of the PLCK-GAPDH mice. When you make an IP of TRAF2, you do find GAPDH bind to it. Oh, sorry, an IP of GAPDH, you do find TRAF2, and the opposite is true. When you make an IP of TRAF2, you do find not only the endogenous, but also the tagged form of GAPDH. So this is a very schematical representation of the NF-kappa B pathway, and the ability of TRAF2 of GAPDH to bind TRAF2 should lead to an activation of NF-kappa B. And this is indeed of the canonical pathway. And this is indeed what we observed in T cells of young mice, where you have an increase in the canonical genes. But it has been shown that a prolonged canonical activation of the canonical pathway can lead to an activation of the non-canonical pathway through different modalities. And indeed, we observed in older mice, when they get sick, that we have not only the canonical, but also the non-canonical pathway that are activated in those thymocytes, in those T cells. So to show that finally this activation of the NF-kappa B pathway was really related to the phenotype of the AITL, we did a couple of experiments, genetic alteration of the NF-kappa B pathways. So the first one was to use the mice model developed by Véronique Imbert and Jean-François Perron in our center, where they overexpressed, they deleted, E-kappa B beta specifically in T cells. So those T cells will have a constitutive activation of the NF-kappa B pathway. And when you age those mice, after 18 months or more, you do see the appearance of T follicular helper and GCBs as we observed in the GAPDH mice, but never observed in the control mice. So to prove our point further, we did another experiment that was to prevent the ability of NF-kappa B to be activated when we overexpress GAPDH. So we created the PLCK GAPDH P65 deficient mice, and we used the thymocyte and differentiated the thymocyte in vitro into T follicular helper. So you can see in wild type, you have some T follicular helper. When you have GAPDH overexpressed, you have way more T follicular helper. But when P65 cannot be present, when NF-kappa B cannot be activated, GAPDH is not sufficient to lead to the conversion of differentiation into TFH. So the NF-kappa B, the non-canonical NF-kappa B pathway is mainly controlled by the kinase NIC, that is upstream of the pathway. And we decided to target, to get interested in this kinase, not only because it's overexpressed in TFH, as we saw, and leading to the activation, participating in the activation on the non-canonical pathway, but also NIC is overexpressed in JCBs. So targeting NIC will allow us to target both cell type that are the main component of this AITL pathology within the same, at the same time. So we had a nice, very fruitful and a nice collaboration with Professor Thierry Passeron and Dr. Rachid Benida, who created the Yukin Therapeutic company to develop NIC inhibitors. And we did the following experiment. From the PLCK GAPDH mice that is sick, we transfer the lymphomas into recipient mice, and we treated the mice with the NIC inhibitor in association or not with PD-1 in order to remove the T follicular helpers. And the results are here. The treatment of with the NIC inhibitor alone or in association with PD-1 was very efficiently protecting the mice. And finally, we could confirm our result in patient sample in collaboration with Dr. Vaida Snafi and Professor Philippe Gollard that sent us primary sample of 5 AITL patients. And most of those patients, when you look at the CD4 and the T cells and the B cells, they were their proliferation were highly impacted in vitro by the use of the NIC inhibitor. So I hope I convinced you that having more GAPDH makes a difference in the T cell lineage, and we turn out to create by accident a novel and very interesting model of AITL that resembles very closely to what is found in patients. And we found also that targeting the NF-kappa B pathway, and actually more specifically the non-canonical pathway of NF-kappa B, could be a valid therapeutic approach, as we observed this huge increase in this NF-kappa B pathway also in patients. So the take-home messages of all this presentation is GAPDH is a very interesting enzyme. I hope it convinced you. And it's interesting for different reasons. Not only it plays a role in metabolism, as we all know, but it also have non-metabolic role, and the threshold between each of them is mainly controlled by the amount of GAPDH present in the cell. And actually, it has been shown that GAPDH is not a limiting enzyme of the glycolytic pathway, but it becomes limiting when you are under the Warburg effect, or when you want to prevent the non-metabolic functions. That is, I think, a very nice target to follow. So I would like to emphasize and to come back on the notion that when you study metabolism in cancers, it's extremely, you should be extremely careful to not limit your work on the use of cell lines only. It's completely okay to use cell lines to start with, but obviously the metabolism of those cell lines is completely different from real life. And if we want our results to be more ready to be transferred to clinic, we need to make a big step towards the use of more proper models. I hope I convinced you that the use of metabolic inhibitors requires pre-selection of the patients. It makes no sense to launch a clinical trial if you cannot predict the metabolic status of a given patient. And now this OXPHOS and glycolytic phenotype of the tumor has been shown in a variety of tumors. It's not only in DLBCL, it's shown in different tumors, including solid tumors. And as we know, as far as we speak, GAPDH is the first and only predictive marker of the metabolic status of a tumor. I showed you that PLCK-GAPDH represents a novel AITL preclinical model that can be used to eventually test molecules for this rare disease, and it's extremely complicated to find and to test molecules in this for this rare pathology. And finally, we could show that targeting the non-canonical pathway of NF-kappa B could represent, we hope, maybe in the near future, a therapeutic option for those patients. So finally, keep in mind that GAPDH is more than a housekeeping gene. And also, I hope I convince you that all the work we tried to do over the last years is, of course, an enormous combination of brainstorming, fundamental research with close collaboration with clinicians, motivated and passionate clinicians in association with companies. And this is exactly what the Institut Carnot-Calim is doing. And they are putting, and all the team, Bertrand and all the team is putting a lot of efforts to favor this interaction between clinicians, companies, and fundamental researchers for, we hope, in the near future, a benefit for the patients. With all that, I would like to conclude there, and I will be happy to take any question you may have.
Thank you, Jean-Ehrland, for this very nice presentation and this very, very nice data. I really enjoy and appreciate this talk. So, yeah, there's a lot of questions, actually. There's a lot of questions. I think that the first question is in the concern the first part of your talk. So you nicely described the DLBCL in actually two groups. Maybe the OXPHOS and the glycolytic DLBCL, or the GAPDH, the GAPDH high and low. And so the first question was, okay, is it really two groups, or can you see some kind of plasticity among the same tumor, some kind of heterogeneity in the metabolism of a patient or tumor?
No, it's absolutely right. If you do, for example, the scoring or the precise metabolic characterization of those cells, it's obviously a continuum between highly glycolytic tumor to highly OXPHOS tumors and everything in between. But that's probably true for all the metabolism. It's not a black and white thing. It's obviously a threshold, and we had to cut, in the...
Sorry, the question was,
if I can go...
Yes, please.
Go ahead.
No, the question was about the cohort, but among a patient, a tumor, one patient, can you see some kind of heterogeneity, or if you are black and white?
No, sorry. Within the same patient, very surprisingly to us, concerning GAPDH staining, it was extremely homogeneous among the section. We have a needle biopsy of the lymph node, and the staining of GAPDH was extremely homogeneous among the same entity. When we look at the mice, within the same tumor, regardless of its location, regardless if it's in a different lymph node located, we do have the same characteristics. Some mice will have glycolytic tumors, some mice will have OXPHOS tumors. We don't have heterogeneity in those models.
Okay.
That's very surprising, because you said yourself that in development, in T-cell development, and probably in B-cell development as well, and of course, depending on many factors, environmental factors, you may have the privacy, and you may have switch from glycolytic to respiratory dependency. Why should it be frozen for tumor? Because that's what your observation is making us to believe.
Well, that's of course the million-dollar question. We don't know why. We observe that it is obviously the case in patients and in mice models. But I do agree with you, metabolism is extremely plastic, and that's what I try to show you at the beginning of my talk with those different connections and tight bounds between the different metabolic pathway. You probably noticed that all the mice, which is not that sad, but also the patients finally relapsed under treatment. So very likely they adapt their metabolism, they switch to another source of carbon or another source of energy within the same tumor entity. And that's actually where we are going to understand how this switch is made, and probably to offer sequential inhibition of those pathways.
Still related to this question, Jean-Ehrland, you made the parallel between the Margaret Sheep classification of OXPHOS and glycolytic classification that was supplanted by the loose classification of ABC versus GCB. But so, do you have a strict relationship between GAPDH and OXPHOS versus glycolytic, or do you have a little bit of low and high in both?
So, of course, it's more robust in mice because we could compare a lot of samples. Because you have to understand that in humans you need fresh samples. I mean, as soon as the sample is frozen, we cannot really analyze easily the metabolism. So we did make a very nice connection, thanks to Catherine and the team in Hôpital Saint-Louis, to receive fresh samples, and we did find that we have a very, very nice correlation in a few patients and in all the mice we have with the amount of GAPDH and the metabolic status of the tumor.
Okay.
Another question, so there's the two groups, the OXPHOS and the glycolytic, and you told us that, there's a poor correlation with the GC, ABC subclassification, but now, there's some kind of description of molecular classification, of molecular subgroups and classification, and can you fit the, the metabolic status with one of the 5 genomic subsets?
So, obviously, we did it extremely extensively because that was our first idea that, let's say, the ABC group would be one of them, a type of phenotype. But of course, we couldn't show that. And we tried really hard in all possible conditions. To be honest with you, we didn't take the more recent classification because it's extremely complicated to make, and especially we need primary human samples that has been characterized to do so. So the short answer to that is ABCGC, no correlation whatsoever. More fine-tuning of subclasses, we don't know.
Okay, and in the same way, I have a stupid question, but LDH is a basic marker for lymphomas in the clinic, and we assume that it's a marker for the tumor mass and or the proliferative status, or maybe the spontaneous lysis. But do you think that it could be a correlation between the LDH, the seroic, the LDH level in the sera, and the metabolic status of the tumor? So
obviously, what you find in the serum, it's what has been released, and as you said, it's obviously a readout of the lysis, of the cell lysis. We didn't find nice correlation between LDH, and neither the response to R-CHOP or the metabolic status. We didn't observe that.
Okay, thank you. And a new question regarding this, after we will move with the second topic, but a last question regarding the difference between the glycolysis and OXPHOS tumors. Did you find some difference in the tumor microenvironment between these two groups of DLBCLs?
No, we didn't actually. At least in the mice model, the lymph node is filled at 95% of B cells, I have to say. So it's extremely homogeneous, and we have little variability. This is probably not what we found in humans, but in the mice model, it's like one cell basically filling all the one type of cells. In humans, in all the sections we analyzed, we didn't find anything striking, I would say.
Okay, perfect. So I think that there are some questions regarding the second part, the AITL story and the GAPDH in T cells. So there was a question. So interestingly, you detect the ROE mutations in the tumors, but did you detect the other canonical mutations of AITL, so STAT2 mutation or DMT3A mutations?
So actually not. We did a genome-wise sequencing of I don't know how many mice, and that cost us a fortune. And the outcome was like none of the other, SDH, TET, and so on mutations were observed in our model. The only one we could observe is the ROA mutation I described to you, and some kind of indel mutation in ROA also.
But was it the exclusive mutation that you found, or although you didn't find the ones that are known in human, you found some new ones that might be maybe related in mice?
Correct. Yeah, that's why we characterize it that it is indeed a loss of function as it is in human. It's a new mutation that leads to the same outcome. And of course, I know it's bearing, it's coming. Why we have ROA mutation? I mean, the short answer is, of course, I don't know. My guess is we have this very early on activation of NF-kappa B that is present within the thymocyte. And the pathology appears like in human, in very old mice, 18 to 20-month-old mice. So my guess is this high inflammatory chronic inflammation in the T cells, in those peripheral organs, will favor the probably, I don't know, random or specific accumulation of ROA that has been shown to be a trigger of this AITL effect.
Yeah. Maybe there is a missing link with the epigenetic, because there's a questions asking that, if you identify or observe the link between metabolism and epigenetic reprogramming or signatures in lymphoma, a link between the metabolism. Yeah.
So as you know, there is an enormous amount of link between the metabolism status and the epigenetic. We know this AITL is, the epigenetic is central in this model. So obviously, this is where we are going towards. We want to understand if the metabolism is different, if for example, generation of oncometabolites, so we don't have SDH mutation, but we could imagine others as being described, and they could somehow impact on the epigenetic of those cells. This is ongoing.
Yes, and there's a question regarding maybe the metabolites. So do you think it could be we could observe some kind of metabolite exchange between the tumor cells and the microenvironment in your models? In
the AITL model, you mean?
Yes.
Yeah, very likely. You know that better than me. The proportion of T and B cells, it's only a small portion of the tumor where we know the microenvironment in this type of lymphoma is central. And this is also a main problem, because when you do, for example, a PDX model, you lose this heterogeneity, you lose the context, and finally it's a poor model of this type of tumor. So obviously there is crosstalk and dialogue. We didn't characterize it. You can imagine it's extremely complex, but very, very likely happening.
Yeah. We can see questions, very different questions regarding the maybe the NIC inhibitions, the NIC inhibitor. So the question is, NIC is known in cancer to be involved both in canonical and non-canonical pathway. And have you checked if which NF-kappa pathway is targeted with your inhibitor in your models?
So we didn't do a very clear characterization of that, but obviously the company that is developing this, the UK company that is developing this inhibitor, has the lead on that, and is doing that very, very carefully in different kind of cancer, as you can imagine.
Yeah, sure.
In this respect, it's my turn to ask a very naive question, because considering the fact that you described the importance of this pathway and the efficiency of the NIC inhibitor towards both GCB cells and TFH cells, might want to foresee that other type of tumors might benefit from those inhibitors, such as the ones that rely highly on TFH, such as follicular lymphoma, for example.
Well, obviously, that's really the field of the company, and believe me, they have a lot of very nice candidates. But unfortunately, we are not allowed to
sure.
on that, as you can imagine. But the idea to target the NIC was really to kill the two main players with the same drug, to kill two birds with one stone. And it turned out to be extremely efficient.
And what about the other inhibitors that you talked about? Do we know that they are acting directly towards the tumor, or can they as well it's probably a much more complex effect toward all the tumor and the microenvironment.
The metabolic inhibitors in the first.
Yes, the metabolic inhibitors. Yes.
Well, you're absolutely right. They are extremely wide and would have an effect in the global metabolism of the patient. It's extremely hard to discriminate what is happening. In the case of L-asparaginase, there is a lot of on this molecule to know what could be the side effect and how we could lead to this very significant effect. And I think the depletion of both amino acids, I don't know which one is the most important, but it's obviously a key event and very promising, I think, in this pathology.
If I may, Francois, I see you about to ask another question, but you described in the beginning of your talk for the DLBCL the sequence of therapeutic actors that are at the moment foreseen for patients that are not responding to treatment. How would you foresee combinations and where in the therapeutic sequence? Seeing that today, DLBCL is very crowded, particularly with CAR-T, but
obviously, my guess will be at the end of what I've been describing with this red cross, at the end of the standard of care. I think there is room for the use of proper metabolic inhibitor in a proper patient. You cannot do that in all of them, but very likely the patient will relapse, as we saw before. But maybe it's a good strategy, for, I don't know, while the core T cell are being prepared for the given patients, or while you can meanwhile analyze the metabolic switch, have another marker of the switch, and offer a new therapeutic inhibitor to those patients. That's probably where I would put the.
But have you checked that something that you look at diagnosis is still true in the sequential relapse?
So unfortunately, in patients, we couldn't do that. We didn't have enough material to test that. We did that, of course, in mice, and we do see adaptation, as you can imagine, and we are characterizing it. But we are, and hopefully we're working with you at Kalim to prove this point in humans, but having, biopsies of the same patient, sequential biopsies at different steps of the treatment is not that frightening.
Okay. So maybe it's time to conclude, Bertrand?
Yes. If we're done with the question, and unfortunately the time is running out, so I would like to thank you, Jean-Ehrland, for doing this really fantastic talk. We enjoyed it a lot, and many questions. Thank you as well to all the participants to have played the game of the webinar. It's a new way of interacting. I hope that you will enjoy the next Redtalks, and of course Jean-Ehrland will be able to look at all your initial questions, and if you have some more questions, please feel free to connect with either us or him directly. But it was a great pleasure to have you with us tonight. Hope everybody enjoyed this first Redtalk. And I have to now say bye-bye. I don't know if I have to say have a good day or have a good evening, but anyway, have a good time.
Thank you to all of you. C'est fini?
C'est fini, je pense qu'on est off the hook. Ça s'est super bien passé. Merci mille fois.
C'est pas fini.
C'est moins agréable que de voir les gens. Hein.
C'est pas fini, donc le...
Bah, c'est sûr, mais on profite quand même. Mais c'est parce que tu n'avais pas ton verre. Tu vois, moi, le mien, il est fini. Hein.
T'as raison. Moi, je suis à l'eau aussi. Bon, en tout cas, merci pour cette
on est encore
plateforme.
là.
Merci à tous. Merci à l'équipe Kalim qui est derrière tout ça. C'était vraiment super pro. I enjoyed it tremendously. Merci et bonne soirée.
Merci beaucoup. Ouais.
Et du coup, tu sais, on peut retrouver les questions quelque part, parce que du coup, juste par curiosité.
Ouais.
On les voit, si je ferme, ça s'enlève? Comment ça se passe?
Ben regarde maintenant, tu peux les regarder maintenant. Je pense qu'elles resteront, mais je suis pas sûr. Donc,
les questions, ouais.
Ok, ben je vais me balader 5 minutes sur le chat.
Ok, ouais, je pense que ça vaut le coup, c'est assez sympa. C'est principalement des questions du consortium, mais néanmoins, ça permet de connecter. Ciao, bonne soirée.
Merci beaucoup.
À bientôt. Merci beaucoup.
Merci. Ciao.